Good evening, everybody. My name is Neil. I go to Adelaide East Stevenson in Illinois. Parnitha and Avantika, do you guys also want to introduce yourselves?
Hi, my name is Parnitha. I'm a junior at Massachusetts Academy for Math and Science. Avantika?
Hi, I'm Avantika. I'm a sophomore at Andover High School and I live in Massachusetts.
All right. So, from pixels to patterns, tracking brain development in mice using image analysis, by Parnitha, Neil, and Avantika. So, what is the brain and why should we study it? The brain is an essential organ that regulates and keeps the body working properly. The brain is responsible for behaviors, motor skills, and it's basically what makes us human. So then why should we study it? By understanding changes in apparently like between apparently healthy patients brains and that of like a patient suffering from, let's say, like a disease, we can understand the regions of the brain responsible for various medical abnormalities. And project goals. So our goal was to gain insight on the development of the brain. And we achieved this by taking measurements of thin slices of the brain, looking specifically for the area of the cortex and hippocampus. Then we put all the measurements of the little slices back together and we grouped them by the age of the mouse to understand the development of the brain across time. For more background, the leftmost image. Thank you, Pranitha. The leftmost image is a representation of our brain, the human brain, and the cortex, and all those different little colors are subsections of the cortex, and adding them all up together is what makes up the whole cortex itself. The middle image is a representation of the hippocampus, especially in the dark blue. That is mainly what the hippocampus is. And then the rightmost image is a representation of the mouse brain since we did look, this research was about a mouse brain and how that compares to a human brain, and that has all the little notations.
So for our data, we looked at the Allen Brain Institute's Mice Brain Atlas, and they had images of coronal sections, which we'll have an image of on the next slide, which were stained for the specific cells, so the cell bodies are visible on there. And they had ages from embryonic day 18.5, right before birth, all the way to P56, which is like basically an adult. And we used Fiji, which is a software that you can outline and get data from. Me, Neil, and Avantika used Fiji separately and then combined our data so that we reduce our bias and account for any human error. So using Excel, we created scatter plots, and these were our results. So here is an image of a P28 mice and section 256 from the Allen Brain Atlas. And here you can see the cortex outlined. So from these analyses, we got a scatter plot going from anterior to posterior, so from the front to the back. And you can see that as as the brain ages, there's more area, and it's not a linear progression. So there's certain periods where the brain develops faster compared to other periods. And it's the same for the perimeter. Then we have the hippocampus, which is really important for the memory. And we have the same image, but with a different part outlined. Again, you can see that from 18.5 to P56 is increasing, and you can see a really high spike right here. Same with the perimeter.
So, through the data, we found a lot of interesting information. Firstly, the cortex appears as an already layered structure at E18.5, and the cortical neurons are actively migrating to their final positions. This means that the brain has developed, this section of the brain has developed quicker, and it's almost done developing, but it's still in the process. And we also noticed that the deeper layers of the cortex are more mature, while the superficial layers on the top are still experiencing growth and development. The cortical surface also appears smooth, and the measurements are markedly small for E18.5 in comparison to postnatal day 56. And by the time the mouse has matured, the cortex has grown into its adult six-layered structure, and, and the measurements are visibly larger as the cortex has reached its full adult size, volume. For the hippocampus, it's basically the same thing. There was a vast difference between the size of the hippocampal region between the embryonic stage and P than the fully matured adult measurements. In the image to the left, you can see this is an example of a mouse brain in the embryonic stage. You can notice that the basic C structure of the hippocampus is present, and the dentate gyrus is still being formed. That's a major subregion in the hippocampus. And the image to the right is an example of a mouse brain that is fully developed. It's in its adult stage. You can notice that the hippocampal region is fully mature and clearly defined, and the area and perimeter is many times larger as there was a large increase in the number of neurons and cells. So this is where we come to our conclusion, why it's important that we study mouse brains. First of all, studying mouse brains is both an ethical and proficient way to provide a foundational understanding of healthy brain development. We specifically use mice because mice genes are around 85% identical to human genes, and their brain structures are very similar to ours. By using this knowledge, we can understand, diagnose, and treat human neurodevelopmental disorders and investigate the impact of genetic mutations and environmental factors. By mapping the trajectory of development between the embryonic stage 18.5 and postnatal day 56, this can for example, neurogenesis, which is the process of generating new neurons within the brain. And other than developmental processes, we can also study diseases or disorders in humans with genetically modified mice. By studying their brains, we can create drug therapies and treatments that can help alleviate these illnesses and help more people in the future.
So for me, I really love neuroscience, so I love this project a lot. I also gained so many skills learning how to use Fiji, creating graphs, and analyzing data. So overall, this was super fun and informational, and I just loved it.
This project was very interesting. Learning about the brain, such as like the neurons and growth cones, was like very insightful. I also like learned new applications such as Fiji. And one thing I liked about Fiji was it was mostly straightforward, and also exploring the applications such as writing code for it and all that. That was also like a fun challenge for me.
For me, I agree. This was a great project. I've always been interested in neuroscience, and I've not really had a support system in my school, but this internship was really perfect to dive into the analysis of the brain with technology and analysis and platforms I haven't used in general.
We would like to thank Dr. Manasi Agarwal for guiding us throughout this internship and
The Horizon Quest program for giving us the opportunity to participate in this internship.
And we would like to thank all of you for coming here and listening to our presentation and supporting us. So thank you.
Thank you, guys. Everyone did very wonderful job, and it looks like you're going to be maybe neuro researcher soon, like by the brain development, the mice brain, and like the human brain, and doing the Physi application. This is the one of the most useful software that we guys use too for even the brain, lungs, everything that you can do with this image analysis. Yeah. Great. I have one question for you guys, and maybe somebody can go after me. When you guys did the like select the sections, like which section I'm going to select, did you choose like anterior, middle section, or posterior, and keep consistent? Like what you guys choose from the coronal sections?
So Dr. Agarwal sent us every 10th section from each of the age groups so that we would have a consistent anterior to posterior.
Mm-hmm. Yeah, that makes sense because you want to be consistent with like whenever you are selecting, if you want to go with the anterior, you want to choose the anterior sections throughout the development from the embryonic, juvenile, or adult brain. So yeah, 10th section. If anyone have any more questions for this wonderful team? Okay, so no. So I am going to move forward with our next presenter.